So non-Mendelian inheritance. That's the topic for today. So we're getting away from the complete dominance problems where we have one allele that's dominant and one that's recessive. And the first type of non-Mendelian inheritance pattern that we're going to look at is this type of pattern known as incomplete dominance. And I'm going to give you a very simple non-textbook definition of incomplete dominance. We're going to define this as a blending of traits. So where this is different from complete dominance is in complete dominance, you have one trait that's dominant and one trait that's recessive. In incomplete dominance, neither trait is dominant. Neither trait is dominant to the other. And so if you read the text or you look at this, there are several different ways to represent alleles in incomplete dominance. We're going to be looking at flower color as the trait or the characteristic. And the way that I'm going to represent it is what I feel like is the easiest way. I'm going to use R to represent red, and I'm going to use W to represent white. Now, the text and a lot of other methods that are used use prime. So they would use R for red and R prime for white. I feel like that gets a little bit confusing. So with incomplete dominance problems, I like to use different letters to represent the different alleles. And that just helps me better keep it straight. But use whatever method works for you. If you like using the same letters and making one of them the letter and one of them the letter with a prime symbol, you are more than welcome to do that. So red and white, I make them both capitalized because neither red or white are dominant in this example. So we're looking at the flower color of snapdragons. What we're going to do is we're going to start with our P generation, just like we did with monohybrid crosses, just like we did with dihybrid crosses, and we're going to carry that P generation all the way to the filial 2 or the F2 generation. So to be able to do that, as you've learned, the parental cross or the P gen has to start out with two individuals that are homozygous for different traits. So we're going to start out with a homozygous red and a homozygous white. Once we have our allele assignment that we did up in the top, once we have our test cross where we do in our P-gen, then we can set up our Punnett square. So keep in mind, those alleles, we separate them. And again, that's Mendel's law of segregation. They are separated or go through what we call disjunction and anaphase of meiosis. And they are separated and then they are individually packaged into their own unique gametes. So just a reminder, on the outside of the Punnett square, those are gametes. And then on the inside of the Punnett square, that's going to represent the possibility or the probability of a fertilization event that would produce a zygote. Same rules apply. Whatever's on the top is going to drop down. Whatever's on the left is going to move over. Another helpful hint here is notice how all of them are the same. I made them all RW. I didn't make one of my boxes RW and the other box WR. Even though that would mean the same thing, it doesn't look the same. So we want consistency with this so that we don't get confused. We don't want to confuse ourselves. A good rule of thumb that you can use is if you're dealing with different letters, whether it be incomplete dominance or codominance that we're going to learn about here in just a little bit, use the alphabet as your reference. So R comes before W in the alphabet. So I'm going to put R first every time while dealing with this incomplete dominance problem. So from this, I get my genotypic ratio and I get my phenotypic ratio. And since this is the generation after the P-gen, this gives me my filial 1 or my F1 generation. Now, if I look at my Punnett square, I can observe that I have an RW in every box. So all of my genotypes in my Punnett square are the same. So I would describe that as all RW. Now, phenotypically, this is where it's going to be a little bit different. Remember up here, we identified incomplete dominance as a blending of traits, and we said that red was not dominant and white was not dominant. Therefore, we get a blending of the two. So this is kind of like being in art class and taking red paint and white paint and mixing it together. And when you do that, what you end up with is pink. So a red and a white make a pink. Okay, so all of these flowers in that F1 generation would be pink. Since we began with two parents up here in our P-gen that were homozygous for different traits, that allows us to get 100% of the same genotype in the F1, which allows us to do our F1 cross. And so our F1 cross would be RW crossed with an RW. And we know that because that is all that we have to cross in that F1 generation. Then I work out my Punnett square just like I always do. The alleles go through segregation and disjunction and anaphase and are packaged into their own individual gametes. Same rules apply. Whatever's on the top drops down. Whatever is on the left moves over, keeping consistency with different letters. So again, my R is always going to come before my W. Now, if I look at my results from this, I have three different genotypes. I have a big R, big R. I have an RW, RW. And I have a WW. So there are three different genotypes here. So when I'm looking at my genotypic ratio, the only ratio that we've established that allows you to have three different things is that 1 to 2 to 1. So I'm going to say that I have 1 big R for every 2 RW for every 1 WW. We've also established that the phenotypic ratio can be different than the genotypic ratio, but in this case, since this is an incomplete dominance problem, it's not going to be different. So this genotype would translate into, I have 1 red for every 2 pink for every 1 white. And this genotypic and phenotypic ratio would be representative of what we would expect to observe in our F2 filial 2 generation. So another type of non-Mendelian inheritance pattern that we're going to look at is going to deal with blood groups. So the next thing that we're going to look at is ABO blood groups. And one of the terms that's associated with ABO blood groups is this term known as multiple alleles. So we say that ABO blood groups are multiple allelic, okay? And what that means, and I'm just going to put a number to it, there are 3 alleles for blood groups. Now, when we say that there are 3 alleles, that means that there are 3 alleles for ABO blood groups within a population. Each individual still only has 2 alleles. So if we were to look at everybody's blood type in the class, then we would, I'm sure, discover that all three of these alleles that I'm getting ready to show you they exist within our population. However, each one of us, each individual, only has two alleles because you only have two parents. Just like every other trait, you get one allele for blood type from mom and one allele for blood type from dad. But there are three alleles that we can have. And so the A allele, we're going to represent as capital I, A. The B allele, we're going to represent as capital I, B. The O allele, we're going to represent as lowercase I. Now, before we begin working out some of these problems, some of these blood type problems, we're going to make a chart, and there's a couple things that I want you to understand about ABO blood groups, some terminology, and some inheritance patterns before we begin working out these problems. And so we're going to start off this chart by looking at the phenotype. And the phenotype, when we're dealing with ABO blood groups, is actually the blood type. Now, this is a little bit different than other traits that we have learned about. And it's different because with other traits, you can look at a flower and tell if it's purple. You can look at a flower and tell if it's red or white or pink. You can look at a pea plant and tell if it's tall or if it's dwarf or if it's yellow or green or round or wrinkled. You can look at those physical characteristics. Blood typing is a little bit different. We cannot look at one another and determine what the blood type each individual has just based off of appearance. So instead of a physical description like purple, round, yellow, wrinkled, green, instead the phenotype is the actual blood type. And so there are 4 different blood types that are possible from these 3 different blood alleles. And this is something that you guys probably already knew, but you can have type A blood, you can have type B blood, you can have blood type AB, or you can have blood type O. So what we want to establish for these different blood types are the homozygous genotypes. We also want to take a look at the heterozygous genotypes. And we also want to establish what type of inheritance pattern we're dealing with. So I'm going to make this into a nice, neat chart here. So for type A, the homozygous genotype, so if we refer back up here to our letter assignment or our allele assignment, a homozygous A would be capital IA, capital IA, or, and I'm going to show you how you can do this a little bit differently, and I'm going to show you this because you're advanced enough to be able to go back and forth and do these types of problems interchangeably. I can tell you if you see a question about ABO blood groups on an in-class exam or on the final in the end, then you will see it represented with the capital I's with the letter of the blood type as a superscript. But you could also simplify this by just saying AA. That would be the homozygous genotype for type A blood. Heterozygous genotype. That would be capital IA, little I. So it's heterozygous, so it's different or mixed. And so you could put capital IA, lowercase I, or you could put AO. The type of inheritance pattern that this is complete dominance. So we're very familiar with this, and what that means is if you have an A allele and an O allele together, you get type A blood. A is completely dominant to O. If we take a look at blood type B, the homozygous genotype would be capital IB, or you could represent that as BB. And we're going to work out a problem in a second, and I'm going to show you how to use either or, and I would recommend that you pick the one that's going to work the best for you. The heterozygous genotype, very similar to what we did with type A. We're going to have either a capital IB, lowercase I, or we can represent that as a B and an O. Just like with A, this inheritance pattern is complete dominance, because when you have an allele for B and an allele for O and you put them together, you still get type B, which means that B is completely dominant to O. Type AB. We are first going to do the heterozygous genotype. So the heterozygous genotype would be capital IA, capital IB, or you could simply say AB. Now, with the homozygous genotype, I'm going to go ahead and X through that because there is not a homozygous genotype. It does not exist for type AB blood. The inheritance pattern is also not complete dominance, nor is it incomplete dominance that we learned about with flower color. This is a new type of dominance that you're learning about, known as codominance. So let's take just a moment to establish how codominance is different from incomplete dominance and how it's different from complete dominance. So codominance is kind of like having co-captains on an athletic team. If you have co-captains on an athletic team, then they share leadership responsibility equally. The same is true here. If you have an allele for type A and an allele for type B, you have type AB blood because they are represented in the phenotype equally. And we'll look later on at what that exactly means and how that translates into the types of proteins that are produced and displayed on those red blood cells. If this were incomplete dominance, then when you put a type A or an A allele and a B allele together, then you would get a completely different type of blood. So if you put an A and a B together, you might get type C. That would be a blending where you didn't get A and you didn't get B. You got a mixture of the two, and that's not the case here. You put an A and you put a B together and you get type AB. Same with incomplete dominance. If it were codominance when we looked at flower color, when we put a red flower and a white flower together, then you would get those equally displayed in the phenotype. So you would get a flower that had a red petal, white petal, red petal, white petal, red petal, white petal, etc. So that's the difference between incomplete, where neither are dominant and you get a blending of those traits, versus codominance, where they're both dominant and they're both equally displayed. And we'll look at other examples of codominance later on besides just blood typing. Blood type O, homozygous genotype. You could represent that as little I, or O, O. Just like type AB did not have a homozygous genotype, type O does not have a heterozygous genotype, because we've already established if O is with anything else, either an A allele or a B allele, then it is masked because A and B are completely dominant to O. So the way that type O is inherited is it is recessive. So these are the things that you need to know and understand in order to work out a blood typing problem. What we're going to do is we're going to work out a blood typing problem using both of those allele representations that I showed you. And we're going to do one that shows as many different possibilities as we can and gives you a new type of ratio. So we're going to cross a heterozygous A with a heterozygous B. So you can represent it like I did on the left, or if you like doing it this other way better. And again, my recommendation would be to pick the one that's going to work best for you, but also to be able to, if you like doing it the way I'm going to do it on the right better, then just be sure that you can go back and forth between those two. So for both, we're going to draw our Punnett square. Same rules apply. We went through disjunction, through the anaphases of meiosis, and those gametes went through the segregation and were packaged into, or those alleles, went through segregation and were packaged into their own unique gametes. Same Punnett square rules apply. Whatever's on the top drops down. We keep A's in front of B's, and we keep dominant alleles in front of recessive alleles for this. Oh, wait a second. Sorry, troll messed up right there. Okay, so you get something like that. Now, if we look at these Punnett squares, you will notice that we have 4 different genotypes. There's not any 2 genotypes in this Punnett that are the same. And so we're up until now, we've dealt with either a 1-to-1, a 1-to-2-to-1, a 3-to-1, an all, or a none. Here we're actually going to have a 1-to-1 to 1-to-1 ratio. So I'm going to look at my genotypes and phenotypes for each one. And over here on the left, I would say that I have 1, capital IA, capital IB, for every 1, capital IB, lowercase I, for every 1, capital IA, lowercase I, for every 1, lowercase I, lowercase I. On the right, I would say that I have 1, AB, for every 1, BO, for every 1, AO, for every 1, OO. So remember that genotypes, whenever we're doing a genotypic ratio, we have to include the ratio, and we also have to include the genotype. And with genotypes, each individual has 2 alleles because they have 2 parents, and they got 1 from the maternal chromosome and 1 from the paternal chromosome. Now, phenotypically, it doesn't matter which method you use, you're going to have the same phenotype. So capital IA, capital IB translates into 1AB. So does this genotype here. Capital IB, lowercase I, translates into 1 type B. BO also translates into 1b. Capital IA, lowercase I, translates into 1a, and so does AO. And then lowercase I translates into, and that kind of looks like a 10, but that's 1O and 1O. So phenotypically, regardless of how you work this out, you should get the same phenotypic results. Really quickly, I'm going to show you how to work out one of these as a dihybrid. And so to do that, and we're not going to talk about what this actually means just yet, but the RH factor, we have RH positive and we have RH negative. And so you hear this positive and negative attached to blood type descriptions all the time, A positive or AB negative, that sort of thing. And like I said, we're not going to go into detail about exactly what this means right just yet, but I do want you to know that the positive is dominant to the negative. So we're going to take a look at this from the perspective of a dihybrid cross. So I'm going to say that I'm going to cross an individual that is AB negative. And so to be AB negative, you have to be homozygous recessive for the RH factor. And we're going to cross that individual with a heterozygous B that is also heterozygous for the RH factor. Now, for dihybrid crosses, we're not going to take the time to work out a full 16-member Punnett square. I'm going to remind you of the shortcut that you guys have learned about. And that shortcut is looking at how many different allele combinations we can have from each parent. So if we look at the parent on the left, we can have a capital IA negative. And it doesn't matter which negative we put that capital IA with, it's still an IA negative, so that's one possibility. Or we can have an IB negative. And again, IB negative or IB negative, those are the same thing as well. So we have two possibilities for this parent. For this parent, we can have an IB positive or an IB negative, or a little I positive, or a little I negative. So we have four possibilities there. So in this example, we're going to have a Punnett square that has 6 squares. So with dihybrid crosses, you can have as few as 1 square in your Punnett, or you can have as many as 16 if both parents had have been heterozygous for both traits, which they were not. So I'm going to draw my Punnett. As a 2 by 4. So I said that this parent on the left has 2 possibilities. So on my Punnett that has space for 2 possibilities, I'm going to put capital IA negative on one and capital IB negative on the other. Across the top here, I'm going to put the 4 possibilities that my second parent can have. One of those would be IB positive, another would be IB negative, another would be little I positive, and then the last but not least, little I negative. Same rules apply. Whatever's on the top drops down. Whatever's on the left moves over. So a typical question, and as we mentioned before, when you're dealing with dihybrid crosses, you're usually asked to work out a pretty complex problem. You're not often asked to do all of the genotypic ratios, but you might be asked something like this. If these two parents were to produce an offspring, what is the probability that they would have a certain type of blood? Okay. And so, for example, if this said, what is the probability that this couple would have a child who had A positive blood? Then you would have to look at this punnet and identify, well, here's type A positive, because this would be A negative, this would be AB negative, AB positive, B positive, B negative, B positive, B negative. Okay. And so we would say that the possibility of them having a child who had um, type A positive blood would be 1 out of 8. Okay. Or if the question said, what is the probability that this couple would have a child that would have B positive? Then right there would be B positive, which would be 2 out of 8, 1/4, or 25%.